Right. So I'm Weng Ming, and I'm currently a postdoc at the Jenner Institute, which is a vaccine institute here at the University of Oxford. First of all, allow me to thank you, Alistair, for introducing me, and MBIOS for inviting me to give this Research Insider talk. And please do accept my apologies in advance if you hear, coughing fits throughout my talk, because I'm actually recovering from a flu at the moment. So I title my talk called My Science Journal Onward. I quite like the word onward because it means that I haven't reached the destination yet, or I hope I shall never reach the destination, because I hope my journey towards the science discovery is always fueled by my passion, my curiosity, and I will never reach the end goal where I feel like I don't know what's next, because there are always more things to discover and to work on. And so I thought I would give very brief introductions about myself. So I was born in 1993 in Damansara in KL. So I'm from Selangor. You don't have to do the maths. Don't do the maths about what my age is. So it's just jump forward to 2000 when I attended my primary school in SJKC Chanmore in Petaling Jaya. And then in 2006, I attended my secondary and high school in Catholic High School. And after 5 years of Form 1 to Form 5, I attended 2 years of A-levels educations in 2011 to 2013 at Chempaka International School in Cheras. And then I literally took a flight on an actual Malaysia airline plane and arrived in London in 2013 to start my undergraduate, where I studied biochemistry at Imperial College London. And after 3 years of undergraduate studies, I started my PhD, or we call DPhil here in Oxford, which lasted about it's got about half a year delay because of COVID. It was about 4 to 4.5 years. And since then, I've been a postdoc here at the Jenner Institute at the University of Oxford. So science, yes, today we're talking about science, and most of us are bioscientists. So maybe I should tell you why I'm interested in science and why I become a scientist. So since I was a little kid, I've been interested in science, and science is really, really broad. It's a very broad term where everything can be categorized into very three vague categories: biology, physics, and chemistry. But there are many, many more subgenres under them. And so when I was a little kid, I was interested in dinosaurs. Actually, I still am very interested in dinosaurs, and that's paleontology. And then when I was a teenager, I was very interested in astronomy, where I studied and read every books about the universe and the galaxy and the stars and the planets. And then when I was in high school, when I studied science, physics is actually my favorite subject out of the three amongst biology and chemistry. Not really because of all the formulas that you can see here on the screen, but it's really more about everything feels sensical and logical, where you throw a ball up in the air and you can calculate the velocity and the time, the force, and out of those things feels really neat and very satisfying. So why now I am doing biosciences and how do I arrive at this location is what I would like to tell you today. So in high school, I'm a science stream student. So in Malaysia, we have, if you still remember in our high school times, we have art streams, which is a little bit more about humanity and business studies. And in science stream, we study biology, chemistry, and physics and mathematics. So it was an easy choice for me to continue on studying science during my A-levels, my 2-year A-level study. But how do I go on to study biochemistry for my undergraduate? How do I decide what course to choose? It's actually not that difficult. I didn't really struggle much in terms of, what I need to study so I can get a job in the future. It's actually quite sort of decided, quote unquote, quite decided for me, because in our A-level system is quite exam-based. So if you look at each of the subject, biology was mostly memorization. So you just have to memorize the the standardized answer, the exam format, you memorize the names and the text, then it is quite easy actually for you to score high. In contrast to, for example, chemistry, physics, and mathematics, which require a little bit more analytical thinking. So, so biology was naturally the highest scoring subject for me. So I thought I should do something in biology, but I had no interest in zoology and ecology and biodiversity and plants. But biology is really general and wide. But I was interested in molecules and the small things and immunology and parasitology and virology. And it's sort of a similar things for chemistry, where I am very interested in small things like organic molecules, but I was not really interested in inorganic and synthetic and computational chemistry. And as I told you, I was really interested in physics and I actually loved it. But at a time of my sort of education, I wasn't really aware of any job prospect as a theoretical physics. So I thought I can do biology, I can do chemistry. So why not I just merge both of these together? And that's why I decided to do biochemistry for my undergraduate. So my take-home message here really is just let your interest guide you. And if you like me, which also like most people have more than one interest, just find a way that can combine as many interests as possible into one thing. And that should guide you down a very successful and very interesting path down for your career. Don't worry too much about jobs prospect really at the stage of the undergraduate, because how many people I know or who really study undergraduate course and went on to utilize that degree for their career or for their job? Not really, because I have many colleagues or friends who studied biochemistry as me with me and end up being a consultant or a work in banking or financial districts, or work in other parts of science or change science. I think so. This is not really should be an obstacle for you to choose what you're interested in. And of course, how do I actually know I want to do what I'm interested in? When you went on and asked people, oh, why do you do this? Or because I'm interested in this, and that's a very vague answer. So I decided to test my interest. So before even I started my undergraduate, so it was between the first and second year of my A-level studies, it was in summer 2011, I decided to reach out to a professor in UCSI University in Cheras, which is about 50 minutes drive away from where I was doing my A-levels. And I was very grateful that she decided to accept me as a summer student for about 2 months there, so I can do a little bit of internship to work in the lab. Because before that, our studies during high schools and during A-levels, our lab experience was really, really, restricted and very minor in the way that we just mix things in test tubes and record colors and write, lab reports. So I wanted to, in a way, step into a real world and do some more expensive experiments. And there was a summer when I was doing some genotyping DNA from different individuals of varying levels of cholesterol. And that's a little bit more genetics, because at that time I was really interested in studying genetics. And after starting my undergraduate studies, and it was actually first year of my undergraduate, summer 2014, I reached out to another professor who actually taught in my course, Dr. Constantino Baez, who I remain very grateful today for his acceptance, my acceptance at his lab during this summer, sort of formed my career in a way to become a structural biologist. So what you can see here is a ring, and this is what we call a synchrotron. It's a very, very huge ring. So it is in this ring where electrons are accelerated to a very, very high speed, close to the speed of light, where at the bands in these rings, there is a slight loss of energy, and this is the energy that is transformed into a very high energy X-ray beam. And these X-ray beams can be used in experiments. And this is the summer when I discovered structural biology when I was working on X-ray crystallography, which I will explain to you a little bit more later. And in summer 2015, which is after the second year of my undergraduate, I decided to see if I'm still interested in what I want to do. If I still want to do lab work, if I still want to do science, stay in academia, if I'm still interested in structural biology. So I was accepted to a summer placement program in Munich. So I went to Technical University of Munich and joined Professor Michael Settler's lab to work on nuclear magnetic resonance, which is a technique quite similar to MRI when you go to a hospital and take an MRI and scan. So this is called NMR. So after a few summer placements where I sort of reaffirmed myself that I'm really interested in science, I want to do lab work, I want to continue being a scientist. So in autumn 2015, I applied for PhD, and in early 2016, I was accepted for a PhD program. And in the same year, I finished my undergraduate and started my PhD in late 2016. So again, my take-home message here is you have things that you're interested in and you want to, and you should test them. You should double confirm it, you should triple confirm it, because your interest can change. It's a bit like hobbies, isn't it? I was interested in playing badminton 2 years ago and now I no longer play it. So you just have to make sure you're still interested and actually have the experience of doing what you're interested in. Because in this way, it's not really good for your CV, for your lab experience. It's really good for your own self to know that you want to do what you want to do. So a question that is commonly asked for me, to me by students, how do you know you want to do PhD? Should I do a PhD? And PhD is difficult, it's scary. Should I ever start a PhD? And it is not an easy job because it's not a 1-year course or a 2-year master. The shortest PhD here is about 3 years in the UK. And if you attended PhD in other countries, for example, in the US, on average, it's about 4 to 6 years. So it's not a thing that you can take without a full commitment. So how do I know I want it? So as you know, I studied biochemistry for my undergraduate, but I also love physics. So I thought, can I, in some way, like I just told you, merge things together? So I can merge biochemistry and biophysics together. So as I told you, after my first year undergraduate, I went to the synchrotron where I was working on X-ray crystallography, where the X-ray beams are generated in these synchrotrons. And this is the first picture I took actually during my summer placement there. And I think this is a picture that I will forever cherish and I will forever keep, because I think it's a very important photo for me that sort of memorialize my passion and my excitement at that time. Because when I walk into the beamline, so beamline is sort of the hutch where the X-ray beams are directed from the synchrotrons, and the X-rays can be used for experiments there. And when I walk into that beamline, and these are the machines that I saw, and I was so excited, not only by the machineries, but by the application of it, by the freshness of it, refreshness of it. So this photo meant something for me, and I took this photo with me everywhere I go, and actually, went on and started a career on structural biology. So really, in this photo, where you can see, if I zoom in a little bit, is where you put a sample here, which is your biological samples in the form of a crystal. This is, it can be a protein crystals, for example. And then the X-ray beams are directed from this hutch here, and then the X-ray beams are shot on these crystals. And then you get a diffraction patterns, and all these dots referring to the samples in these crystals where it was hit by the X-ray beams and diffracted onto the detectors, where you cannot see on this far left-hand side here. And then through mathematical and biophysical models, you can transform these dots into actual atomic models, a 3-dimensional atomic models. And this is very, very interesting for me, and this is what we call a structural biology. So I'm a structural biologist. So what is a structural biology? Structural biology is a branch of molecular biology, biochemistry, and biophysics, all combined, concerned with 3D molecular structure. So we use structural biology to obtain 3D molecular structure of biological molecules, such as proteins, RNA, DNA, membranes, virus, bacteria, and a lot more, and how they acquire the structures that they have, and how these structures look like, and how these structures inform us their function, how the changes in this structure change their function, and how we can interrupt this structure, whether to gain of function or loss of function. So this is what interests us, not only for structural biologists, but for across the entire biology field, because these molecules carry out functions in cells. And we want to, for example, in where I work in General Institute, which is a vaccine institute, we want to look at how we can use these structures to make vaccines. And so here comes another reaffirmation to myself. So why do you like structural biology? So you say you're interested in biochemistry and biophysics, so you decided to do structural biology. So it is not a very interesting answer. It is also very vague, because biochemistry plus biophysics can lead to many other fields, not necessarily structural biology. So it is a question that I've asked myself for throughout my entire undergraduate degree, until I met a professor in Rockefeller University when I was there, because I was accepted for a PhD program there. And it was during a dinner when we were all sort of chit-chatting and hanging out. So a professor sat next to me, was asking me, oh, what your hobbies are. And I was like, oh, I was a dancer for several years. And she was like, oh, me too. And we were really excited about talking about art and dancing. And then the next question she asked me was, oh, why do you like structural biology? And I struggled actually to come up with an answer that I feel satisfying. So I just told her, oh, because, it gives me pretty pictures. And she agreed. I thought that was quite a little bit of a lame answer, but she agreed. And she said, yes, that makes sense, because you're, structural biologist, she is a structural biologist as well. And it's sort of a way, sort of in tune with our artistic sense, and we dance and we like arts. It's a little bit like that. And I thought, yeah, that makes sense. And since then, I stuck with that answer. And I think it is a very true answer, at least for me, that I do love pretty pictures. And I love seeing the structures, and that structures tells me some things. In opposed to reading something with just charts and graph and lines and bar charts, pictures, pretty diagrams are what interests me. From proteins here to viruses here, which, some of the ones that were most popular now and most recognizable now, which is SARS-CoV-2, which caused a COVID pandemic, to what's inside the cell. For example, a double nuclear membrane, to all the vesicles and ribosomes and whatnot in the cells. So these give us pretty pictures, and they're not pretty pictures for pretty pictures' sake. They're also important to tell us, like I just told you, their functions and how we can, create therapeutics and prophylactics against diseases and infections that we're studying. So I use structural biology to study viruses. So I'm a structural virology. So many people have used structural biology to study some other things. For example, some other people specialize in membrane proteins. Some people specialize in studying bacteria, certain bacteria. Some people specialize in studying ion channels, and I used to study viruses. And here comes another questions. So people ask me, why study viruses? And this was actually a question that was posed to me at a conference in Madeira, when I went to Madeira in Portugal. It was actually a professor that I met there, and we later become friends. And he said to me during, sort of a drinking session after a long day of conference then, and he said, quick, give me your elevator pitch. Why study viruses? And the first answer that came into my mind was, why? Because I love a challenge. Because I always think humanity, man and virus are always trying to outsmart each other. So I think it's a competition, and I like a challenge. So this diagram here on the right is sort of a very popular figure now, especially at the beginning of the pandemic, which illustrates, sort of the pandemics that we have had since the beginning of records, ranging from the highest death toll to the lowest. And a lot of these are actually caused by viruses. And you can see this is smallpox virus from HIV to flu, a lot of influenza viruses to smallpox. We have yellow fever, we have COVID-19, which obviously now we have more, way higher death tolls. And then you can see a trend here. From the beginning, it was mostly pox viruses, and we have influenza viruses. But on and on, towards recent times, you see new viruses coming up. Ebola, MERS, SARS, and even the ones that are not here, Zika. And we know there are a lot more viruses that cause epidemics, not necessarily pandemic, but epidemics like dengue virus, chikungunya virus, Lassa virus. And all of these viruses do cause quite a significant amount of deaths. So in some way, they're not exotic, but you can say that they're the viruses that cause human diseases and severe human diseases are becoming more and more exotic, quote unquote, and become more varied. So I am interested in this way that we have efficient vaccines for measles and polio and smallpox and mumps, and in some way we have eradicated some of these, but we have no efficient vaccines for many of the diseases, viral infections that we are facing these days. So I want to use structural biology to study, to understand the mechanisms, the infection mechanisms of these viruses and how they infect hosts, especially humans, and can we intervene with this infection process? And can we actually intervene? And that's the application, biology and application. So as I told you, my structural biology uses X-ray crystallography as one of the techniques, which uses this high energy X-ray beam to obtain the structures of biological components. And another technique I use is cryogenic electron microscope, which is quite similar, somewhat similar to X-ray crystallography in terms of the mathematical models, but instead of X-ray beam, we use electrons. So this is sort of like a 2.5 meter tall box, and inside you have many metal machines, and inside this machine there's a very big vacuum chamber where high energy electron beams are directed and focused towards your cryo samples, your frozen samples, and that's sort of a similar way you get your structures. And I use structural biology to study viruses, from Lowy River virus, which is a novel virus discovered in Thailand in 2016, I believe, to Hunin virus, which caused hemorrhagic fever in Argentina, Chupu virus, which caused hemorrhagic fever in Bolivia, to Lassa virus, which caused constant outbreaks in West Africa. All of these have high fatality rates, up to 30%. And down to some of the ones that we recognize, for example, dengue virus, which plagues Asia, Africa, and South America, same as Zika virus and chikungunya virus. And all of these are moving away, not moving away, but sort of expands out from Asia and Africa continents towards regions where it was previously cold because of global warming. For example, it's now moving towards North America and certain parts of Europe. And of course, for a while, I was working on SARS-CoV-2, and I'm now working on rabies virus, and I'm looking forward to work on the next virus as well. So I thought I'm going to give you some flavors of how I use structural biology to study viruses and how it informs us certain important things about these viruses. And there's nothing in depth here. It's just a little bit of brief introductions to structural virology. So here, I use structure to inform vector selection. So we have collaborators who went to Brazil to study chikungunya virus, which is a virus that is transmitted by mosquitoes. And they realized that, So the chikungunya virus are mostly transmitted by Aedes aegypti in Brazil. There is a preferential for this species, for this lineage of mosquito. So why? So decided to collect more than 75 virus genomes across Brazil and do high throughput generation sequencing and identify unique positive selections. So these are the mutations, they are preferred, they are positively selected. And as structural biologists, we then use this information to map back on the structures, and we realize that one of the mutations is lysine. So this is an amino acid residue lysine 211, which has mutated to glutamate residues here, prefers Aedes aegypti, and we realize that it is because this is the residues which sits at the interface, which interacts with the receptor that the virus used to infect the cell, infect the host. So disruption of this interaction between the virus protein and receptor will also modulate, will also affect the entry of the virus into the cells. And in some way, this sort of hypothesize why the virus in Brazil, due to these positive selections, favors Aedes aegypti rather than Aedes albopictus in some other areas. We also use structural biology to understand virus evolution. So in this tree is what we call a structure-based phylogenetic analysis. What you just need to know is the longer the length, the less related these viruses are. So there are different families of virus, or they're the same family, but they are not so related. So in here, they are all arenaviruses, so they're within the same family, but within the same family, we realize they are quite different. So for example, here, we found out that Lassa virus, LASV, actually have same protein, but actually have two structures. So it's the same protein, but it presents to the cell two different structures. And we were confused about why it has two different structures. And combined with some of the medical journals that we've, literatures that we found, we realized that it could be because one of these structures is actually an immunological decoy, where it was either secreted or as a byproduct during the virus infection, where it was presented to the antibody producing B cell, and then the antibodies would then target and recognize this structure, which is actually a decoy, rather than able to recognize the actual structure that are presented by the virus itself. So in some way, you cannot, there are always virus infection that cause a lot of antibodies, but these antibodies are actually ineffective against the virus infection. They do not clear the infection. So this is one of the ways that the virus evade the immune system. And this is also later on found out or discovered in some other viruses as well. For example, in HIV and Ebola, where they also have proteins which adopts different structures as immunological decoy. And here we use structure to understand how the virus are neutralized by antibodies. As I mentioned before, Hunin virus, which caused hemorrhagic fever in Argentina, and Machupu virus, which caused hemorrhagic fever in Bolivia, they have case fatality rate up to 30%, but yet there are no internationally approved therapeutics. So we know antibodies are very powerful. They can neutralize. So we want to isolate some of these antibodies, and we want not only to isolate them, we want to understand how they neutralize and if they're useful to use as therapeutics. So we decided to take this virus and we immunize, or we vaccinate mice, and then we isolate their splenocytes to obtain B cells. And through a series of experiments called single B cell sortings and so on, we obtain these antibodies. And we bind these antibodies to the viral protein, and we use structural biology to obtain the structures. And you can see that this antibody here bind to a groove on the viral protein. And it is this groove, exactly this groove, that the virus used to bind to the receptor to enter the to enter the cell that the virus want to infect. And you can imagine that if the antibodies already bind to these pockets and hence exclude or preclude this receptor binding, prevent this virus able to bind to the receptor, it will then stop the infection process. We also use structural biology to study glycan functions. So a lot of the proteins presented on the surface on the virus are glycoproteins, which means they are proteins which have sugars, we call glycans, attached to them. And these glycans have functions. We want to start, so we use structural biology to understand the functions of these glycans. For example, here, we see that this is just a protein. This is not a virus. This is just a small protein on the virus surface. And these are all these colorful chains that you can see kind of protruding out from the protein surface are the glycan chains. And you can see how dense they are, sort of entirely wrapping around the entire protein molecule. And this is what we call a glycan shield, where they're really protecting the protein surface from being detected or being approached by our immune cells. So even antibodies are generated, they are very difficult to reach within these pockets, these small spaces, because they are so densely covered or shielded by these glycans. Another virus tell us where the receptor or rationalize where receptor and antibodies can bind because of how the glycans are formed. You can see the glycans are formed around a pocket, and it is these pockets where the receptors and the antibodies are recognized. We also can rationalize how this is an entire virus. Now this is dengue and Zika virus, and glycans can also be used to bind receptors. So here we can see that there are different kinds of glycans presented by dengue and Zika, which then tell us dengue and Zika can use different types or utilize different types of receptors to enter a cell. And here we use structural biology to tell us how we can design vaccine or design immunogens for vaccine purposes. So here's rabies virus, which cause encephalitis, which are almost always 100% fatality once symptoms appear. And we do already have a rabies vaccine, but it requires 3 to 4 doses. And if you're if let's say you got a rabies dog bite, you need to get about 3 to 4 vaccine doses, and you need to get also immunoglobulin, which is antibodies treatment. And all of these cost a lot of money. They are very expensive, and not many people have the money, or not a lot of countries have a lot of these therapeutics available because demand exceeds supply. So we want to create a better vaccines or design a better vaccines. So we use structural biology to obtain the structure of the rabies glycoprotein, which then tell us where the antibodies can bind. And not only that, it tells us how the structures interact with one another and how we can stabilize this structure in a way that they are easy to be produced in the lab or in the factory or in the manufacture plant. And so we can lower the cost of vaccine. We can use this structure to tell us how we can change it to become more potent, or you can elicit a higher neutralization antibody titer in trials and all of that informations. And of course, just a quick slide on SARS-CoV-2, which structural biology definitely helps, is using structural biology to understand mutations. So we know that now we have many variants from alpha to beta to delta Gamma to Omicron now, and now we have different variants from Omicrons, and they all have many mutations. And due to selection pressure, virus can mutate inside the infected host. So we use structural biology to understand how some of these mutations can escape neutralization, and how we can design, use structural biology to design better immunogens or design better antibodies that can target these mutations. So that's the flavor of science, that I do, which I just told you, how I harvest the sort of the various interests in my science career and combine to become structural biology, and reaffirm myself with various lab experience and questions to make sure I am truly interested in structural biology. And I use structural biology in my career to study virology. And I'm a postdoc here, so I'm part of the academia. So science are always split in, well, generally speaking, are split into academia and industry. That's some people like to say, whether you work in a pharmaceutical company or you work in science communications or and so on and so forth. So I'm part of the academia life, and people always think, oh, it's just a laboratory, which means you just work at the labs all day. It's not necessarily true, because I would say a very important part of academia is networking and collaborations, because no one can do science all by themselves. So, I went to all these conferences or symposia, attend workshops, to increase your skills, and seminars, to give talks, to talk about your work, to, connect with collaborators across the world. So I have attended many conferences. These are just some of them here, and made many friends, and made collaborations. And it's truly wonderful times, actually, really, to start a career in academia. So with that, I end my talk. Of course, I like to thank MBIOS again for inviting work and use of Oxford at the General Institute within the Department of Medicine. And I thank many of the funding partners. With that, I end my talk. Thank you. I will take questions.
Thank you, Dr. Wang, for the talk. It's really insightful and interesting. So we have some questions from the audience. So the first question is, going back to the meta topic of SARS-CoV-2, what are your thoughts on its future as a structural biologist?
Structural biology on SARS-CoV-2 itself. So SARS-CoV-2 is now, actually, let's not just talk about SARS-CoV-2. Let's talk about the whole field itself as coronavirus field. I think it is pretty much studied. So the coronavirus produces many proteins, from structural protein to non-structural proteins, to many other, for example, is polymerases and stuff. Structures of all of these proteins are almost studied and produced, apart from a few non-structural proteins that haven't been studied yet, which are currently being studied by a lot of the labs in the world, because everybody now in the world wants to study coronaviruses. I would say the importance of coronavirus in the future might lie less on structural biology, but lie less more on identifications of novel strains of coronavirus, which are a lot of the labs are going into right now, because coronavirus is not just beta coronavirus, which is one of the subgroups in coronavirus. There are also alpha and gamma coronaviruses. And I think these are the ones that people also need to start looking into, because there will be more and more novel viruses, what we call emerging viruses, that have the potential, maybe not yet, but have the potential that jump from hosts, including humans, and then cause diseases.
Thank you for that, Dr. Wang. So another question is, So like the question is, the person is wondering, once you find structural conformation, do you also consider affinity and avidity?
Yes. So structural biology is of course just one of the many techniques that we utilize in understanding a biological systems or biological molecules, as we say. Structures tell us functions and all that. We also use a lot of standard biochemical or biophysical assays like SPR, Biocore, and ITC, and a lot of the things that we study in undergraduates or techniques that we study in our lectures are actually utilized in our labs.
So we have a question from Yi Min. So the question is, what do you find to be most challenging for your current project? What is your most favorite technique at the moment?
My most favorite technique is definitely cryo-EM, which I show you about the 2.5 meter tall box where the electrons are used, high energy electron beams are used. And that's a boom really in the past 10 years, I would say, due to the advancement of technology in creation of direct electron detectors. So better equipped. So structural biology is quite dependent on the equipments really. So better equipments leads to better resolution, which means we can see the structures at a better resolution, means better, clearer stuff. The clearer it is, which means the more finer, the finer details we can see. So that's ongoing big advancement at the moment for cryo-EM fields. As for structural biology, what is challenging? I find a lot of things challenging. So for example, rabies virus, which is my current project that we just worked on, a lot of the biological system that we worked on, they are not very straightforward. We can't just say we express a protein and then we study the structures, because a lot of the biological systems are unstable once you take them out of this, for example, a cellular context. So we then have to spend months and sometimes years to try to stabilize the system stable enough for us to study them in solution. So so a lot of the times go into preparing the samples and understanding the biology behind it.
Okay, thank you. So yeah, so because like like you mentioned that like taking it out of the context of the biologist can be like, can be quite difficult. So like someone is wondering if like in the initial stages of developing a treatment in the context of viruses, do you work together with translational scientists to bring therapeutics out?
So we don't necessarily work in parallel for translational work. So what is the power of structural biology is we solve the structures, we understand how it works, the mechanisms of which the proteins or the virus or whatever you're studying works, and or you design an inhibitor or a drugs or an antibody which targets these specific systems. Now you know how it works. That is all good on paper, in solution, out of the system, but how does it really work in vivo, which is in animals, in the organism itself? We take this information, then we do it in clinical trials, which is what the Jenner Institutes are doing. So we have to do all this understanding of biology first before we can then go on to clinical trials, because we can't just start the clinical trial saying that, oh, I want to test this system, let's do it, because you need to apply through all these paperworks and very, very stringent and very strict, guidelines in order to start the process. You can't just say, oh, I want to have 5 people and come to do this, take these drugs. You need to have all these things established first and understandings of the biology established first. Now, of course, clinical trials are very, very expensive. You can't just with the click of a hand and you want to start. So usually we do the biology first, we have a firm understanding of the biology, and then we start the clinical process.
Yeah, thank you. That's very interesting. So there's another question. So how would the study of structural virology inform other non-virology fields, such as cancer therapy, or maybe novel ways of medication administration by viral injection?
Yeah, structural, well, a lot of viruses do cause cancers. So in some way, you can always link viruses to cancer together. But structural, as I mentioned, structural biology aren't necessarily just used for studying viruses. That's my specialty. A lot of people use structural biology to study membrane proteins, and membrane proteins is about 70% of what we create drugs for. They target the membrane proteins, for example, GPC or G-couple protein, couple receptors. They are the ones that we target because they carry out so much functions, and mutations or the malfunction of these membrane proteins often lead to the malfunctions or misfiring of the signaling cascade, which then leads to cancer or cancer-related diseases. So structural biologists are definitely very useful in understanding these topics.
Okay, thank you. So so there's a question about, like, I guess your career trajectory is, so like someone asked, like, since you're a very qualified scientist, do you have any plans to run your own lab one day? And so what will be the research that you would like to conduct?
Yeah, as I'm in academia, as of current, I wouldn't want to project myself 20 years in the future. That's difficult to tell. But as of now, I very much still want to stay in academia. And I would very much love to start my own lab one day. And my research interest is always more about emerging viruses. So remember the chart when I told you the history of sort of the pandemics which have causes a high death toll rates. So and I make a remark where there are more and more exotic, quote unquote, exotic viruses which cause a lot of human diseases, rather than in the past with just a pox virus and influenza viruses. And coronavirus is a very solid example of that. So I have spent majority of my PhD and now in my postdoc looking at these emerging viruses, which if I mention the names, people will know. For example, now currently I would love to look at viruses, which viruses that cause almost 100% fatality rate, but it doesn't cause a high transmission but high fatality rate. So a lot of these viruses do have the potentials to cause, big epidemic, not necessarily pandemic, but it can cause big endemic or epidemic. And these are the viruses that are worth looking. Not everyone needs to look at the famous one like HIV and Ebola and Zika, because there are thousands of labs looking at those at the moment. So if I ever start my own lab, and actually, I think this is where the virology field is going towards, starting to look at more novel viruses and identify more novel viruses, and clearly characterizing their potentials of causing diseases in humans.
Oh, thank you for the sharing. So there's a question from Clarence. So in Malaysia, the biologists, or like at least the field itself, for biologists or for people who graduate from biological sciences, is limited and underpaid. So would you say it is a phenomenon unique to Malaysia, or it is something that happens around the world, at least from your experience?
Science has always been one of the, I would say, subject, if I can call that a word, say that that is rather underfunded in a lot of countries. So Malaysia is not the only country that is sort of underfunding science. I think it's very much dependent on where the trajectory of the country development is going. So when I was working in the labs in science, or when I was looking into the prospects of science careers in Malaysia, a lot of the the fields going towards more towards translational or application based, which makes sense because Malaysia requires that knowledge transformations of knowledge into applications or applied science, which is why biotechnology is actually I would say more fruitful in Malaysia compared to some of the other countries here, for example, here in Europe, where in Europe we are really more towards understanding the biology. And so we can study more fundamental biology that are not necessarily beneficial to transforming in short-term wise, transforming into a product or increasing a yield of crops and plants and things like that, which are very important. But so this is very important to, I think, for individuals to assess the way the country is moving towards. And of course, you can be a pioneer as well to get the funding if you can, to prove that what you're doing is important. So for example, here in the Western countries, to show that you're studying something in biology, often we do relate to why we're studying them. We need to justify why we're studying them. Most of the time, of course, to satisfy our own curiosity because they are that interesting. We are scientists, first and foremost. But to get that funding, it's not an easy job because it is very competitive, very, very competitive. Most of the funding programs that you see, they fund about 5 to less than 10% of their entire application pool. So you need to justify, by studying this, I actually inform, hopefully, in my case, future vaccine design or importance for translational work and things like that. So for my case, I said it that way. A lot of people use bioscience to study a lot of other different topics. You just have to understand the implication and the justifications of your field and how you can use that to obtain fundings.
Thank you. So from Clarence again, as she is interested in virology, so how would you look for a suitable supervisor to continue PhD? Like, do you have any advice that you can share?
Yeah, so many years ago now. So when I first started my PhD, before I started my PhD, I actually just sent out emails. So one of the things that if I ever take away from my entire science career, you just go and talk to people, get help, write, talk. You will realize that most people are very, very willing to help and guide and talk. In fact, a lot of the opportunities that come to my way aren't because I go and get the opportunities. In some way, it's just through conversation with some people. As people say, oh, I'm working on this. And I was like, oh, I'm actually very interested in that. Can I jump on that? Or, I asked about something through emails to someone, and then someone say, you know what, I will link you up with someone else. So these are the way that you can really get help. So for PhD wise, it's very helpful to just look online first. What are the labs that interest you mildly to wildly, and then write an email and say, hey, I am looking I'm looking towards starting a PhD in this university that you are now have a lab in, and I look at your projects and read some of your papers. They are very interesting. Can I come and speak to you, about the projects in your lab, and perhaps maybe, doing a PhD with you, stuff like that. Yeah. And then go and talk face to face or via Zoom or in any way you like.
Thank you. So, we have like 2 more questions, but I have a personal question. It's like, so because in your presentation, I think I've shared like you did dancing and then you did badminton. So I was just wondering in what ways this, like maybe your pastime, your hobbies have been helpful in your journey in science, which I believe is quite long. And it took you to many locations, so it can be like, maybe it can be quite others. So yeah, just wondering like in what ways your pastime have been helpful in that.
I mean, people always talk about work-life balance, isn't it? And I don't conform to that, because work is part of life. So it is all about life balance, if I would say that. I don't like people saying work-life balance as in, oh, okay, I work 9 to 5 and then I go home, there's no more work. It's not that way. I would say work is part of life. So when I go to all these conferences in the name of work, obviously, it is also a chance for you to, like I said, one of the major reasons you do the conferences is not only to learn stuff from other people or impart your scientific discovery to other people. Its major reason is to really network, to connect people. I've met people who say, you know what, next time if you want a job, send me an email. I literally have professors who told me that. Or second, go and explore the countries that you visit and have fun, traveling. If traveling is one of your hobbies, it is really busy working in science, especially in academia, where even though we do have a 9 to 5 work time, but it's really flexible, where I feel like I work 24/7, not as in work 24/7 on the desk, but my brain is always thinking about science, isn't it? So make it your life, but make it enjoyable. So you have colleagues at work. It's not just about coming into work or your student friends. It's just about I only meet you at university, or I only meet you at the company, right? Make it make them your friends if they're happy to go for I like hiking. We go for hiking together and all these things. Any bondings is great for science, isn't it?
Yeah. So it's already two. Will you be fine with answering the last two questions?
Yeah, sure.
Okay. So there's a question. It's probably out of topic. So what is your perspective on patents law? Do you think there is a need to remove it to help with the pandemic?
Oh, that is a very difficult topic. I have no understanding in patent law at all. I think it is a very difficult thing to say. For example, is patent important? That is the first question we ask, right? Before we come to all these pandemic things. It is important because it protects intellectual property. So whether from TV media or book that you publish down to the scientific discovery that we made, we should protect the intellectual property. But when it comes to use for general good, it is difficult to say because it is very much down to the discoverer. It down to most of the times a lot of these drugs are discovered by companies rather than by academic institutions, and companies, or let's say pharmaceutical companies, are businesses. I think sometimes we have to be less naive and really just actually understand science is political, and science can be, not necessary, but can be business, and it's a large part of science is business. So all these pharmaceutical companies, as we call them companies, they are businesses, and that's why they are profit-based. If you ask me if the world would be better if we just remove the patents and drop the price of the drugs and reach all the people, I 100% agree. But towards the ethics of it and towards the implication behind it, I'm afraid I can't discuss more because it's beyond my understanding and of the scopes of it. Yeah.
Okay, thank you. So we have two last two questions, and it's from Chris. So the first one is like, how do you think the hype on famous viruses like HIV affect research on other neglected viruses that cause health issue as well? And also, I think this is quite it will be quite helpful. Is finally has being part of an like racial minority in England and especially in Oxford, a hindrance in your career or in your journey in science?
Right. First of all, yes. So obviously the popular viruses, for example, HIV and so on, and now obviously SARS-CoV-2 or coronavirus in general, they I wouldn't say hinder other virus research, but they do attract, or they're easier to attract funding because people know them, they know their names. Everyone here know HIV and Ebola and Zika and all these famous names, and they're easier to attract funding. They're easy, like I told you before. Every time we want to study something, it's easier to attract funding if we justify it, or because it's important for vaccine design and so on and so forth. So we understand that it's easier to justify them, so it's easier to get funding. So a large proportion of fundings go to them, and hence funding is limited. So there will be less amount of fundings that go to study these emerging viruses. But I think there's this consensus really in the biology field that is shifting, where emerging viruses really do constitute a large part of future endemic, epidemic, and pandemic, and some of the recent fundings do reflect that, where there are fundings that go on study viruses that I've never heard before and stuff like that, yeah, or related viruses which can then be used to study or create vaccines for the more highly pathogenic viruses. Yeah. And as in being a racial minority in the UK, have I ever received racial remarks in the UK? Yes, of course I have, as do I believe a lot of minors here have experienced. But I would say science, as for me, so far has not been a big impact, not in a way that hinder my career.
So I think I would like to clarify. So is it also an advantage in any ways? Because
yeah, I have never seen that being an advantage. No.
Yeah.
At least not in my case, or not in the institutions that I have been. I can't speak for everyone, but not in the cases where I have been, where they say, "Oh, I have a quota and I must fit how many people." For example, when I went to the US, when I went to Yale's, and when I was speaking to the graduate school admission team there, and during dinner, it was again another friendly chat, and they were like, "Oh, we actually only can accept about less than 3% international students among our application pool," and I was literally the only international people there among like 40 people that they invited, along another Canadian, so just two of us among every other United States citizens. So I think it's not an advantage, but at least in my case, it has not been a hindrance or disadvantage either.
Okay. So I think that's all we have. Questions? Yep. So I think we can end the session. So thank you again to Dr. Weng Ming for coming on a Saturday to share with your experience and for answering the questions. And thank you also for those who came to this session. So for those who have joined late or missed the session, please do check our MBIOS YouTube page for a recording of today's session, which will be published later on. So yeah, thank you again.
Thank you, Alistair. Thank you, MBIOS, for inviting.
So yeah, so that's it. So have a good day, everyone.
Thank you, everyone.